Hello, my name is Adolfo Garcia-Sastre, and I'm going to be talking to you today about some of our work on SARS-CoV-2 biology with an emphasis in our search for new antivirals. I'm professor at the Department of Microbiology and director of the Global Health and Emerging Pathogens Institute at the Icahn School of Medicine at Mount Sinai. And what you are seeing here in the beginning of my presentation, these are cells that are infected with SARS-CoV-2. They have been stained, and there is one stained cell with an antibody against N of SARS-CoV. So you see the infected cell, and with an antibody against in green against AC2, you see the expression of AC2, and these are viro cells. It's an image taken by Mustafa and Lisa here at Mount Sinai. Now, I start my presentation with an influenza slide, and the reason is because most of my work has been centered on influenza, but also what my lab is doing right now in terms of SARS-CoV-2 has to do in part with what we learned from working with influenza. So influenza is also a respiratory virus like SARS-CoV-2. It has also an envelope and a receptor binding protein, which in this case the hemagglutinin. And it's different from the SARS-CoV-2 because of the proteins are obviously antigenically different, receptor is different, and the genetic material is RNA, but in the case of influenza, is a segmented RNA virus with 8 negative strand RNA virus segments, while SARS-CoV-2 is a coronavirus, and it's a positive strand RNA virus with a single RNA segment. Now, in the case of influenza, each of the 8 RNA segments encodes for 1 or 2 proteins, and some of them are structural, as depicted here in the scheme of the virus, and some of them are non-structural, as also depicted here in this slide. Now, if we go to SARS-CoV-2, this is a coronavirus, so this is now a single segmented RNA virus with an RNA genome that is positive strand, meaning that it's going to be used as messenger RNA when it's been delivered inside the cell, and it's going to encode right away from the ribosomes the viral proteins, which is different from the negative strand RNA virus like influenza. It's an enveloped virus like influenza, and the major glycoprotein that is outside of the virion is the spike protein, which is the target for neutralizing antibodies. But it has also another minor components of the envelope, like the E protein and the M protein. And now inside of this envelope, there is the RNA segment, which is a unique RNA molecule that is covered with a viral protein that is the N protein. So when this RNA molecule is delivered inside an infected cell, it's being used as messenger RNA, and it's been encode all the viral proteins. Now, the first messenger RNA that is the genome encodes for a big polyprotein called ORF1A and ORF1B that is cleaved into a total of 16 different non-structural proteins, NSP1, 2, till NSP16. In addition, from the genome, during replication, the virus generates subgenomic RNA that gives another messenger RNAs that now encode for the structural proteins S, E, M, and N, as well as for accessory factors like the ORF3A, 3B, 6, 7A, 7B, 8, 9B, 9C, and 10. And then if you compare different coronaviruses, they have more or less some conserved areas, but the ones that are less conserved are the one for these accessory factors. They are very unique and distinguish different coronaviruses. So the accessory factors of SARS-CoV-2, they are also very unique. Some of them are similar to the ones of the SARS coronavirus, the first SARS that we got a while ago. But some of them are very unique and only found in this SARS-CoV-2. Now, getting back to influenza, this slide is just to illustrate that the virus enters inside a cell, to enter inside a cell in order to replicate, make viral proteins, and make new virions. Now, during this process, the virus has very small amount of genetic material, only 8 genes in the case of influenza, but the virus is gonna need to use host factors that are required for the replication and trafficking of the viral proteins and assembly of the new virions. And in addition, it's gonna encounter antiviral factors that are produced by the cell that somehow sense that it's been infected, and it's gonna try to prevent this infection. Now, in order to find these required host factors and to avoid the activity of these antiviral factors, the viral proteins that are being encoded by the virus need to interact either functionally or physically with these required host factors to help them in replication, or with these antiviral factors to prevent the antiviral activity. And it's in this interplay between the interaction between the viral components and the host components, where at the end it gives you a productive infection or not, depending on how these interactions take place. You're going to get the virus taking over the cell and making copies of itself, or you're going to get the cell being able to avoid and prevent viral infection. Now, with coronaviruses, it's very similar, except that the replication is a little bit different, and these viruses don't use the nucleus, like is the case of influenza, but what it enters through a receptor-mediated entry. In this case, in the case of SARS-CoV-2, is the human AC2 protein. Then it's going to deliver the RNA inside the cytoplasm, and it's going to make some genomic RNAs. And as I mentioned, for the long messenger RNAs, you get this big polyprotein that encodes the replicase and all of the NSPs. And from the others, you get these other type of proteins, including the spike and some of the ORFs. In this case, the nomenclature for these proteins is based in the mouse hepatitis virus, but it's a similar replication cycle for SARS-CoV-2. And at one moment, this virus, like the influenza, needs to interact with host factors that are required for helping in the replication, and it needs also to deal with antiviral factors. And again, like influenza, it's going to be the interplay between the viral components and the host factors, where it's going to, at the end, give a productive infection, more virions being made, or an antiviral state of the cell preventing the viral replication. Now, one of the questions that my lab has been trying to understand in terms of viruses, and especially RNA viruses, and as I mentioned before, mainly with influenza, is which host factors are important for virus replication and pathogenesis. What are these required host factors that the virus needs to use, and what are these antiviral factors that the virus needs to avoid, and that at the end determine what is the outcome of viral replication. In order to answer that for influenza, we have a large program that is funded by NIAID in the form of a U19 grant that we call Fluomics. These are many of the people that are involved in this program with the emails of the people. And it has an intercollaboration between multiple institutions, including my lab, and many people. And what we're trying to do is we try to use a systems biology type of approach to understand the host factors that are important for virus replication and pathogenesis, with the idea of, if we know these factors, we may also know the way how we can inhibit viral replication by inhibiting the host factors that are required for viral replication, or by enhancing the activity of antiviral factors that are trying to prevent viral replication. Now, here is just one example of the type of studies that we do in this program. Here in yellow, in circles, are the different proteins of influenza. You see MP, NEP, and AHA. These are different proteins of influenza. And then they're connected through lines to host factors. And these are host factors that we identified in collaboration with Nevan Krogan as host factors that interact with these viral proteins. So there is many host factors that interact with these viral proteins, but there is a total of 30,000 genes, and this is just a subset now of the genes, of the proteins, of the host that now we have identified as interacting with the viral proteins. And we hypothesize that these proteins that are able to interact, host proteins that are able to interact with viral proteins, are going to be important for viral replication. But in addition, we have here more information, not just the host proteins that interact with a single influenza virus protein, with each of the single influenza virus proteins, but we have also, according to this color code, factors that if you silence them, so if you eliminate the expression from the host cell, are going to result in an impact in viral replication. And depending on the color, there are factors that have been identified by multiple labs or by a single lab. So what we have here now is a collection of host factors that have multiple evidence that they participate in viral replication, either because they interact with the viral protein or because when you eliminate this factor from the host cell, you make an impact in viral replication. Viral replication goes up or goes down. And there is, as an example, there are two host factors that are here indicated, SEC61 and TOP1, that are included among this map. And they are known inhibitors of these factors. CT8 will inhibit SEC61, camptothecin will inhibit TOP1. And we have used these inhibitors to find that they are able to prevent influenza virus replication, or they're able to prevent influenza virus disease in animal model. And I include here two of the papers where we have described this thing. So we are using this map in order to find out novel inhibitors of influenza virus that may be able to use in the clinic. So we have been working on that for several years, and then SARS coronavirus came, the pandemic. And then we have basically used the same resources that they were using for influenza and tried to apply it for the search of antivirals against COVID-19, against SARS coronavirus 2. And it has been, again, a collaboration between multiple people. I will mention some of the main people that have been involved in this collaboration, but there are some people that I will not mention that have also participated. Now, how we are looking for a therapy, and it's not only us, but many people, for SARS coronavirus 2. Now, we know already that there are some compounds that have been approved for emergency use. Remdesivir is an inhibitor of the RNA-dependent RNA polymerase of the virus. Plasma from convalescent individuals, they have antibodies that are able to prevent, by binding to the spike, replication of the virus, binding of the virus to the receptor. Dexamethasone is an immunomodulator, is an immunosuppressor that reduces the exacerbated immune response that happen in some cases of severe infection. And all of these factors have been shown to have some efficacy, some ability to ameliorate disease, but this doesn't happen in all the patients, and they are also not completely effective, 100% effective. So we still need some new antivirals. And in order to do that, the strategy that we're using, we call it drug repurposing, and it's not only us that we're doing that, but there are many people that are also doing that. What is drug repurposing? This is using the already existing drugs that have been used in the clinic in humans. So we know some about what is the effect, the impact of these drugs, what are the side adverse events, what are the targets, for what indications have been used. And what we take is this list of drugs, and then based on prediction, based on omics studies, we make the hypothesis of which of these drugs are going to likely to make an impact in viral replication. And I will show you the type of omics studies and how we do these predictions in a moment. And now once that we make these predictions, then we test these drugs for inhibition of replication in tissue culture. And then if some of them are able to inhibit the viral replication, then these ones we are moving it into animal models. And if they are working well in animal models, then we hopefully will be able to move in clinical studies. But in some instances, actually, some of the drugs that we have found are already in clinical studies for SARS coronavirus 2. The advantage of doing this drug repurposing is that these drugs are known already what is the dose that is tolerated in humans, and therefore one can rapidly deploy it in order to make clinical trials, even sometimes before you know what is the impact in animal models. So this accelerates a lot the possibility of drug development. So the question here is, can we identify compounds that inhibit host factors involved in SARS coronavirus 2 replication among non-human drugs? So first, how we identify these host factors involved in SARS coronavirus 2 replication? So again, we're using an omics approach, and this is one of the ways how we do it. This is a collaboration with Nevan Krogan that is here depicted in this picture. as the source of the host genes, around 20,000 genes that we humans express. And then here the idea is SARS-CoV-2 has a total of 29 encoded proteins. And Evan Krogan has expressed each one individually in cells, human cells, which have all these 20,000 proteins, pellet it down and then determine by mass spectrometry what type of host proteins interact with these viral proteins. And doing that, you create this map that is here in the middle, which is called a virus host interactome, in which in red circles, they are depicted each of the 29 proteins of SARS-CoV-2, and then with lines, they are indicated the host proteins that have been identified as interacting with this one. With the idea, again, that if they interact with the viral protein, probably they are doing something during viral replication. It's being co-opted by the viral protein. So in total, he identified a total of 332 protein-protein interactions. Now, interesting thing is that if you look to these proteins, these host proteins, there are some factors that are druggable, and there are known drugs that are targeting these factors that have been used at one moment in the clinic, or they have been using actually for some specific indications in humans. So here come Kevin Shokat and Brian Soitet, which are our also two collaborators that are chemical bioinformaticians that have looked to this map of protein-protein interactions and then identified among the host factors that interact with viral proteins drugs that are targeting specifically one of these host factors, which is what is included here in this very busy slide. And then they identified a total of 97 drugs. And then in our lab, Chris White, and with the help of Lisa Miorin and also Elena Moreno, have been testing these drugs for ability to inhibit viral replication. And we have some hits, meaning that we have some inhibitors of viral replications that were identified. So how we test these inhibitors of viral replication? For that, we developed a medium throughput assay to look for inhibitors of viral replication in vitro. We use in the beginning Viro6 cells, which they support very well viral replication. And we seed them into 96 well plates. And then after seed them, 2 hours before we infect them, we treat the cells with a drug, and one of the drugs that is known to interact with one of these host factors, and different dilutions of this drug. Then 2 hours later, we add SARS-CoV-2, and we use an amount of infectious viruses that will infect approximately around 10% of the cells, a little bit less than 10%. We collaborate with a group in Paris that are performing similar studies just to compare data. And then we let the virus replicate for 48 hours. And then finishing these 48 hours, we fix the cells, we inactivate the virus, we take this out of the BSL-3. Till now we do these experiments in BSL-3 because it has an infectious SARS-CoV-2. And then we determine the extent of viral replication. So if we have an inhibitor of viral replication, the virus will have replicated less. Now, how we look for viral replication? In our lab in New York, we use 2 assays. The first one is the rapid assay, in which we take the cells that have been fixed and we stain with an antibody against one of the SARS proteins. So if there is no inhibition of viral replication, approximately 80% of the cells have been infected after these 48 hours in contact of cells and viruses. But if there is an inhibitor of viral replication, there is less cells that will be infected. In addition, the ones that are given a hit, we take the supernatant from these cells, and when we determine the amount of viruses that have been produced in the supernatant as tissue culture infectious dose 50s. And again, if we have an inhibitor of viral replication, then you will get also less viruses. In addition, we conduct one more assay, which is not shown in here, which is a cytotoxicity assay of the compound in the absence of virus in the presence of cells. So making sure that if we get inhibition of viral replication, is not because the compound is killing the cells and then therefore the cells cannot support viral replication, is because the compound has direct antiviral activity. And our collaborators at the Institut Pasteur of Paris in France, Olivier, Marco, and Christophe, they conduct very similar assays, except that instead of using a staining against MP for determining the amount of infection, they use a quantitative RT-PCR to quantify viral genomes that are being replicated in the presence or absence of the inhibitor. And if they are inhibiting, they perform plaque assays to find the amount of viruses that are being present in the supernatant. And this, by the way, how a plaque assay looks like for this virus. This, what you see here, are viro cells monolayers that have been infected at different dilutions with SARS-CoV-2. And then after 48 hours, if there is a lot of viruses, then the cells are being killed, and therefore, if you stain the monolayer, you will not see a blue color. And as you go in diluting the virus, then you are starting to see these holes that have been made in the monolayer. These holes have been made by one virus particle that infects first the cell in the center of the hole and then propagates to the next one and killing them. And that's how we can titrate efficiently the extent of replication of the virus in the supernatant of these cells. Okay, so first I'm going to show you how remdesivir, which is a drug that is already approved, inhibits viral replication in this assay. So what we have is in all of these assays of inhibition of viral replication, we perform triplicates in which a particular drug is used at a total of 6 different concentrations from around 10 micromolar going down to nanomolar concentrations with 6 different dilutions. And then what you can see is that in the discontinuous line, you see the cytotoxicity of the drug at different concentrations, which in the case of remdesivir is not cytotoxic. The cells tolerate well remdesivir. That's good news. And in the solid line, you see a extent of viral replication. And what you can see is that as you use increased concentrations of remdesivir, you are inhibiting viral replication. With the ones that are high levels of remdesivir, you don't detect infected cells. At low levels of remdesivir, you detect the same percentage of infected cells that in the absence of remdesivir. And this allows us to calculate what we call IC90, which is a inhibitory concentration 90, in which the compound is inhibiting 90% of viral replication, which in the case of remdesivir and for viro cells is 1.5 macromolar. And also what is the selective index, which is how close is this inhibitory concentration to cytotoxicity, which in the case of remdesivir is more than 26. So it's a drug that can be used for inhibition of viral replication. Now, not everything that inhibits viral replication in vitro is going to work in vivo, but some of the things that inhibit viral replication in vitro are also going to work in vivo. And these are the ones, this is the first assay that we do to see whether we get an inhibitor. Now, you do the same assay, not in viro cells, which are monkey cells, for remdesivir in our human cells, and for that we need to, this human cell, we need to express AC2 because does not express AC2 to allow the virus to replicate. So these are done now in 293 T cells, AC2. Now, actually, the IC90 of remdesivir go from 1 macromolar into 24 nanomolar, which is more characteristic of what is the effective dose of remdesivir in humans. The reason why it's more effective in human cells, remdesivir, than in viro cells, because remdesivir is a prodrug that needs to be transformed into the real drug once that you give it. And the enzymes that transform the prodrug into drug, they are more active in human cells than in monkey cells like viro cells. And that's the reason why remdesivir is more active in human cells. And that's also the reason why we normally use more than one cell type in order to make sure that an inhibitor is not only cell type specific and also works in human cells. So now we have taken all these compounds that they were predicted to inhibit one of these host factors, and we have conducted all these antiviral assays. Here is an example of some of the ones that we've done. We have done now more than 400 tags, not only coming from this study, but also coming from another studies. And we have identified some that seems to be very potent in terms of inhibition of viral replication. Not all of them were able to inhibit viral replication, but some of them were very potent. Now, in the first study that we did, which was in the interactome, we found 10 agents that showed good efficacy in killing viruses. We get the same results as the results that they get with the same drugs in the Institut Pasteur of Paris. And among these drugs, we found 2 different types of compounds, some compounds that are inhibiting translation of proteins, which is sototyphin and ternatin-4, and some compounds that are modulators of the activity of the sigma receptors, sigma-1 or sigma-2, which correspond to a large different type of compounds with different chemical structures, with different indications, but all of them, they have in common that they target the sigma receptor factors, and they have antiviral activity against SARS-CoV-2. So these are the drugs that compounds that bind and inhibit sigma-1 and sigma-2. The reason why we concentrate in these drugs is because we found two proteins of the virus that interact with these receptors, ORF9C and NSP6. And here are some examples of these drugs and how they work. Some work better than others. Interestingly, hydroxychloroquine, which is one of the ones that has been given a lot of attention as potential inhibitor, is a drug that inhibits viral replication in vitro, and it's a drug that also targets sigma receptors. But among all of these drugs, we have some antihistamines, antipsychotics, antimalarials, some hormones, some anti-anxiety drugs, some preclinical drugs. Among all of these drugs, one of the ones that is more potent is this PB22, which is here in the upper right corner, which has a better inhibitory activity than the hydroxychloroquine, and therefore we think that can be better than hydroxychloroquine for inhibiting of viral replication, but that's something that we still need to look into animal models. So one of the advantages of using PB22 versus chloroquine or hydroxychloroquine is that if you look for binding of these compounds to the sigma receptors and to another close receptor, which is a receptor that is found in the heart, ERG, and that is believed that this gives some of the toxicity of hydroxychloroquine and chloroquine, both hydroxychloroquine and chloroquine bind quite efficiently to the human ERG receptor, meaning that it's likely that they have some cardiovascular toxicity, and they bind more poorly to the sigma receptors. However, PB22 binds poorly to the human ERG, so it's likely to have less cardiovascular problems, but it binds very good to sigma-1 and sigma-2, which are the factors that we want to target. So we think that PB22 could be a better compound than hydroxychloroquine, and actually just look for antiviral activity of the compound. It has a better antiviral activity than hydroxychloroquine, so it's improved something like 20 times in terms of inhibitory concentration 90 when you compare both of them. The other set of compounds that we found are these translational inhibitors. So sototyphin is one of them that we are now moving into animal models. It has a target one factor involved in translation, which is EIF4A. The advantage is that has been already in clinical studies for phase 1, phase 2 studies for cancer. It has a very reasonable IC90, around 154 nanomolar, so sub-micromolar. And it has also a relatively good selective index. It looks like there is some cytotoxicity. You see this discontinuous line going from 100% to around 50%, but it stays at 50%. And it means that the compound is not cytotoxic, but it's cytostatic, meaning because these are growing cells, when you put the compound into the media, the cells stop growing. So 48 hours later, you look to what is 100% of cells. In the case of the cells without compound, the cells have duplicated. The cells with compound, they don't grow well. They have not duplicated, and then it looks like you have 50% less cells, which is what the cytotoxicity assay measures, the number of live cells. So this reduction to 50% is not due to killing of the cells, it's just due to inhibition of the growth of these cells, which that means that hopefully will be tolerable in vivo. Now, a similar compound to this thing is this other compound, which is plitidepsin. So plitidepsin is a compound that is targeting this factor EEIF1A. It's a compound that has been developed by a pharmaceutical company, PharmaMar, as an inhibitor of myeloma, and is approved for treatment of myeloma in a couple of countries. And this is one of the compounds that is more effective that we have seen. So you see here an IC90 of close to 1 nanomolar, which is improved even versus the IC90 of remdesivir. Again, remdesivir is better in human cells, but in human cells, this is in monkey cells. In human cells, remdesivir has an IC90 of around 10 nanomolar, and plitidepsin has an IC90 of around 1 nanomolar, so much better inhibitor than remdesivir. This is also looking for the activity of this factor in human cells, not in monkey cells, and again, it has an IC90 that is sub-nanomolar. So it seems to be a very good inhibitor. It's one that is now being used in clinical trials, phase 1 clinical trials in Spain, as well as some have been initiated also in Korea. And we are starting also to be used in animal models to see how good this is working. So this is what we have obtained from the interactome. But in addition, we have conducted more screens also based on systems biology. So here, the next screen that we conducted also in collaboration with Nevan Krogan is based on looking to the phosphoproteome in virus-infected cells. So what is the phosphoproteome in virus-infected cells? These are proteins that change phosphorylation, host proteins that change phosphorylation during infection with the virus. And again, we speculate that if they change phosphorylation, they are going to make an impact in viral replication, either because they inhibit viral replication or because that's something that the virus wants, and then increase viral replication. And knowing the proteins that become phosphorylated during viral infection, we can also predict what kinases are the most likely that are participating in these phosphorylation events. And now what we are going to target are now these predicted kinases that are predicted to phosphorylate host proteins that become phosphorylated in viral infection. We're going to target these host kinases with some known inhibitors. Okay, so here are some known inhibitors from some of the kinases that we identified that have been activated during viral infection. And then this one example, apilimod is one inhibitor of this kinase PIKA5 and has a very good level of inhibition of viral replication. Dinaciclib is an inhibitor of CDKs, which are some of the ones that also we identified that they have been activated during viral replication. Again, it's a good inhibitor of viral replication. We have also inhibitors of MAP kinases that we also identified as activated during viral replication. That seems to be good inhibitors of viral replication. And also in one of these, in the H panel, if you treat cells with an siRNA, meaning you down-modulate this particular kinase, you also make an impact in viral replication. So either down-modulation the kinase by siRNA or by an inhibitor of these kinases is able to inhibit viral replication. So, and the last kinase inhibitor that I'm going to show is this one, silmitasartib, that inhibits casein kinase 2. That is interesting because it's not only activated during viral infection, but it's also known to interact with the M protein of the virus. So we think that has multiple sides of evidence that is required for viral replication. And in fact, it has also a relatively good IC50, around 2.3 micromolar. Okay, but some of our studies have been done not with these omics approaches, but something also like predicting from the structural studies. So this is the case, for example, of a study that we have conducted together with Kuzbu Bafna and Gaetano Monteleoni at the Rensselaer Polytechnic Institute, and also in collaboration with Bob Crook at University of Texas of Austin, in which they have compared the crystal structure of the hepatitis C virus protease and the MPRO, which is a protease that is encoded by SARS-CoV-2 and that is required for viral replication. And even though they belong to different class of proteases, they have a very similar active site. So we predicted that some of the inhibitors of Hep C protease that have been used for treatment of Hep C infections will also inhibit SARS-CoV-2 main protease. And then we tested that in antiviral assays, again, Chris White and Lisa Murin, and Elena Cordero and Romel, now in my lab. And we found that even though they have IC90s that are around 20 micromolar, many of the hepatitis C virus inhibitors like bosprevir or grasoprevir are able also to inhibit SARS-CoV-2 replication. This is another example with faviprevir and simprevir. I just showed you the ones that are working. We found that some others are not working, but these at least are working. And interestingly, one of the interesting things, even though these are inhibitory at macromolar concentrations, if you use in combination with remdesivir, now you can use less amount of these in order to inhibit efficiently. So that means that these protease inhibitors, in this case grasoprevir, is synergizing with remdesivir in terms of inhibition of viral replication. And this one can be seen in what is called an isobol drug graphic, which is here shown, in which you see you put the concentration of remdesivir at IC90, the concentration of grasoprevir at IC90, and giving one constant and the other variable. You put both together. They are more efficient when you put them both together in inhibited viral replication than separately. And so in theory, if you use combination therapy, you are going to get a better inhibition of viral replication than if you use just single therapy. And this makes sense because remdesivir is an inhibitor of virus RNA replication. That means that it's going to be less virus RNA. And if there's less virus RNA, there is also going to be less protease present in the cell. So you're going to require also less of an inhibitor of the viral protease for inhibiting. So it makes sense that inhibitions of RNA polymerase like remdesivir synergize with inhibitors of the protease of the virus. Okay, so I'm going to finish here, and I told you a summary of what we have been doing. And again, this is an example of what my lab has been doing, but many other labs have been also conducting similar screen assays. So it's very, very interesting to see that some of these inhibitors are now coming in clinical trials. First of all, the SARS coronavirus 2 interactome and phosphoproteome provides with rational therapeutic targets for drug repurposing. We have identified inhibitors of sigma receptors, of translational factors, and of protein kinases as potent inhibitors of SARS-CoV-2 replication in vitro. Also, I've shown you that some of the ACV protease inhibitors inhibit SARS-CoV-2 at micromolar concentrations and synergize with remdesivir. But further clinical and preclinical studies are warranted. And I think that one important thing is to consider combined therapies. And I'm just going to thank my lab in one picture that we have just before the pandemic of COVID-19. So that's the reason why we are here without masks. Now we make the same picture. We will be all wearing a mask and doing more social distancing. And I want to also acknowledge not only my lab, but many members of the Department of Microbiology, of investigators in Mount Sinai, other collaborators. I mentioned Nevan and Kevin and Brian and this Pasteur. Some of the patients that we have been studying, although I have not saw here any data with patients, but these are also some studies that we do. And obviously we are very grateful to patients that give us samples for us to study pathogenesis of SARS-CoV-2, and then funding from NIAID, from DOD, from DARPA, and from some philanthropic donations like from OPP or the JBP. So thanks a lot, and I stop here.